My name is Krish Patel. I'm a medical oncologist, and I'm the director of the Lymphoma Research Program at Sarah Cannon Research Institute. I've been in practice for over a decade caring for patients with lymphomas and utilizing the kind of full spectrum of treatments for lymphomas, including cellular therapies, immunotherapies, targeted therapies, and have been an active clinical investigator in those areas for the last decade. So we've seen a lot of exciting things in large B-cell lymphoma in the past few years. So frontline therapy has changed to incorporate antibody drug conjugates into frontline chemotherapy. That's helping hopefully to cure more patients, but we know that a substantial portion of patients will still need treatments beyond frontline therapy. And so what's really exciting in the last few years has been how cellular therapy, specifically autologous CAR T-cell therapy, has improved outcomes in that setting. And so one of the themes I think at ASCO this year was to really build on how are we taking what is a very good therapy already, a CD19 CAR T-cell in the second line, and trying to elevate that further to achieve hopefully more cures for patients who need second therapies or later for diffuse large B-cell lymphoma. So CD19 is a protein that's found on normal B cells and is also found on almost all lymphomas, including diffuse large B-cell lymphoma. So it gives us a target that we can use to essentially target immune cells to attack the lymphoma. To provide some context, I mentioned we have CD19 targeting CAR T cells. CAR T cells are a type of treatment where we basically take a patient's cells from the immune system and then engineer a way for those cells to kind of find CD19 expressing lymphoma and attack them. And those therapies that are available today give us approximately 35 to 40% of patients can be cured with those therapies. One of the things we know that is a limitation for those treatments is we're targeting just one protein in the lymphoma. And so if the lymphoma loses that protein, these therapies may not be as effective. So the study that I'm talking about or will be presenting this summer, J&J 90014496, that's a mouthful, is a CAR T cell that targets more than one protein on the surface. It targets CD19, which existing CAR T cells target, but it also targets a protein called CD20, which is also expressed across almost all B-cell lymphomas. So we get kind of two methods of attacking the lymphoma, and hopefully, that will lead to improvements in outcomes. So we presented data from a phase 1b study of the CAR T cell I described. This was a study that enrolled patients that had relapsed or refractory large B-cell lymphoma. These patients had had 2 or more prior therapies, or for patients who are not eligible for treatment like autologous stem cell transplant, 1 prior line of treatment. And essentially, these patients underwent the usual process for CAR T manufacture and then bridging therapy if they needed it, and then ultimately received lymphodepletion and the investigational CAR T cell at a variety of doses. Some were weight-based, some were fixed doses, and we presented data on the patients that have evaluable results to date. To kind of summarize, most of the patients who were treated that we presented data on are older adults. These were all patients that had some form of large B-cell lymphoma. And these were majority patients that had had 1 prior line of therapy in the overall cohort. And many of these patients had high-risk features like a high IPI score or enlarged tumor burden. These were mostly patients who had advanced stage disease. And the purpose of this study was really first to help us identify the safety of the therapy and to help us define the dose that we might want to carry forward in subsequent trials. And so to talk a little bit about that, what we saw is cytokine release syndrome, which is a toxicity associated with all CAR T-cell therapies. Was common, but it was primarily low grade and manageable. And so that was an important part of the presentation to highlight. Also, immune effector cell associated neurologic toxicity, or so-called ICANS, which is also associated with CAR T cells, was found to be quite low in all comers. So that's, I think, another really important aspect of the study is that we found that with this CAR T cell, the incidence of all grade ICANS was very low, and the incidence of high grade ICANS was also very low, less than 10% for both. And so Moving on to how well the CAR T cell worked, what we saw is across the board, whether you had one prior line of therapy or more, whether you had a weight-based dose or you had a fixed dose of the CAR T cell, as was done in the later parts of the trial, the response rates for the CAR T cell were very high, really 85+% in all comers, approaching 90+% in the majority of patients. And importantly, these were very deep responses with 75+% of patients achieving a complete response, which is the type of response that historically in large B-cell lymphoma translates to good long-term outcomes. So very, very exciting initial data. The patients that we presented also, we had some patients who have been followed on this study for some time, so we're able to speak a little bit about the durability of responses and very encouraged by the activity there. So I think the take home is that J&J 900-14496, the CD19 CD20 CAR T cell, is highly active in relapsed refractory DLBCL, looks very promising, looks to have an excellent safety profile, and there's more experience to be gained with this CAR T cell, but looks to be, I think, a very important potential advance on existing CAR T cells.